Hello, everybody, and welcome to the last poster session for the clinical track. I'm glad to introduce Pauline Trainard, Dr. Pauline Trainard. She is an applications manager at Simulations Plus, and she'll present her poster on count data model for Alzheimer disease progression MMSE score using MonolixSuite. Go ahead, Pauline.
Thank you very much, Jean-Paul. So hello, everyone. In this poster, I present a case study of a count model that we have run in Monolix. And actually, we'll also take the opportunity of this presentation to show not only this poster, but also a glimpse of the model directly in the interface of Monolix so that you can better see what has been done. So I will first show the introduction here. So Alzheimer's disease progression is usually measured with a score derived from answers to multiple questions. And the names of common scores are, for example, ADAS-Cog, CDR-SB, and MMSE. And progression models aim at modeling such scores, usually as a continuous variable, which is actually inconvenient to simulate clinical trials as the continuous simulated values have to be discretized. So in this poster, instead, we model a dataset with records of a MMSE score, which is a test composed of 30 questions, but we consider it as a bounded count data instead of a continuous data. And we model it in Monolix, which allows to easily model any type of data, either continuous or categorical, as well as events or counts in this case. So as you will see, it is easy to implement a custom model by writing the desired distribution. But in addition, since we have done this work, we have also implemented a library of count models within Monolix, which makes it even easier to model such data. Before showing the model, a few more words that you can see here on the data set. That I will use we use for this case study. So the data come from the ADNI study. So it's Alzheimer's Disease Neuroimaging Initiative. The data set is available on the website of ADNI. So here we have 895 patients that were examined every 6 months over 2 years. And we also have some covariates like age, weight, height, and so on. And in particular, also the number of alleles in apolipoprotein E type 4, so APOE. So the questions that we will investigate while modeling this data set are the following. Which model describes best the disease progression and which covariates are predictors of the progression? And finally, here you can see a plot showing the data. I will actually show this plot in Monolix so that you can more easily visualize the data. So this is this plot here. It is a bar plot of the score values across time. You can see that there were 6 visits in total. And at each visit, we have the distribution of scores. So with the smallest recorded value on the left. So for the first visit, it is 18. And the highest value on the right, so 30. And we can notice that most values are high on the first visit with a narrow distribution. Then at each visit, the scores tend to decrease more and more, and the distribution gets wider. And another thing that we can notice is that the number of records seems to decrease as time goes by, which we can see more easily if I choose a stacked version of the bar plot, where we see very well that there is an important dropout with only about 200 individuals left for the last visit. And finally, notice that Monolix provides also a continuous version of this plot that we can see here, which can be interesting to see individual trajectories as well as the distributions for each visit. So now let's come back to the poster. And I will have a look at this part of the poster for the model formulation. So we will see the model used to capture this data. So at each visit, the distribution of score values is modeled with a binomial distribution representing the number of correct answers. So here the number of correct answers is K to the test, and the test has a total number of questions of N, which is fixed to 30. And this probability distribution depends on the probability P, small p, of having a correct answer for each question. And this probability depends on time, and it decreases as the Alzheimer's disease progresses. So we have tested several models to describe the evolution of P over time, and you can see these models here. sorry. So these models have different shapes. We have the linear, exponential, logistic, and Richard models. And so these models depend usually on two parameters, P0 and alpha, except the Richard model, which also depends on the third parameter, beta. And now let's have a look at the implementation in Mlistran, the language to define models in Monolix. So here is the implementation for example, the logistic model. So we have the two parameters in input of the model, then the equation describing the probability P of success, which is here. And then on this line in purple, you see the definition of a random count variable named MMSE, where we define the log of the probability of having k correct answers. We define the log instead of directly the probability in order to use log factorials and avoid numerical errors that could be due to the factorial getting too high, but it is really equivalent to the equation that I have shown here on the poster. So this is a model that we have used for this case study. If you want to use your own model, you can find the syntax corresponding to count models, such as this one on our online documentation, mlistran.lixof.com. And in addition, as I have mentioned in introduction, we have implemented a library of count models in Monolix. So a convenient way to define a count model would be to start from a model from the library, and if necessary, just modify the part of the equation that you want to be different. So let me show you very quickly this library. Here it is available in structural model, and if I load from the library, I can choose the count library, and here you can select a distribution among all this list of distributions that allows to model different types of count data. So for example, binomial is appropriate for a score based on a list of questions or tests, so like in this case study, while a Poisson distribution is appropriate for a number of events, for example. And the other distributions are also appropriate for other situations. So here I could choose a binomial distribution. Then it occurs frequently in count data that there is an excess of 0 counts that are generated by a separate process from the count values. And in that case, the zero inflation option takes care of this property. Here for this case study, it's not the case. So it would not be needed. And finally, you can see here the list of functions available to describe the time evolution of the distribution. So the library is restricted to simple functions, so it does not include the logistic or Richard functions that you have seen on the poster. However, to define a logistic model, it would be difficult to start to load a simpler model, such as a linear model, for example. So I could open it here. I see it here, and then I can open it in the editor. And then in the editor, it would be easy to change the equation here by the logistic equation, for example, and then save this model and load it in Monolix. And so as the rest of the model is already defined with the correct syntax, in particular the variable count number, so the random count variable, it is very convenient to define a model this way, and it is very fast. So this is what we have done for the 4 models that we wanted to test, and I will come back again to the poster to show the results that are here. So after estimating each model, we have compared their results to the estimated parameters, the standard errors, the diagnostic plots, and also the log likelihood, and in particular the BIC, which indicates that the logistic model captures the data best in this case. So this was the first step of selecting the best model. And then after selecting this model, we have used the method SCM and COSAC, which are the different strategies implemented in Monolix for automated covariate search to find predictive covariates. And both these methods gave the same results that are shown in this table, which allows to explain part of the inter-individual variability in the parameters of the model with these covariates. So this was the second aim of this work. The model is also validated thanks to the estimated parameter values that have good RSEs, and also by the diagnostic plots generated by Monolix such as the categorical VPC. So I will zoom a little bit more on the categorical VPC so you can see it well. It compares the observed and predicted probability of getting a score in different interval of values that you can see here on the y-axis. So here is the probability of having a low score, and here's the probability of having a high score, for example. So it decreases over time. So this is one diagnostic plot that is available. And for example, we also have diagnostic plots characterizing the residuals over time, such as this one, which looks quite correct. So this has validated this model. And finally, we have also used, I will zoom on the last part of the poster here, the simulations part. So the disease progression model can be used to simulate the disease progression of new populations with Simulix, which is the application of the MonolixSuite for clinical trial simulations. Here we have simulated 3 populations of individuals with different number of APOE alleles to see how this would affect the evolution of the probability for the score over time. And we have score probability in each group that you can see here. So we can see that this covariate affects mostly the baseline score, as you can see here, and also a little bit the speed of the progression, so the slope in this plot. So this is an example of how you can simulate very easily a new clinical trial with this kind of count model that allows to really simulate discrete variables that are completely adequate compared to the data that we have modeled. And this concludes my presentation of this poster.
Thank you, Pauline. This was great. Do we have questions? Not yet. So let's launch a poll in the meantime, and otherwise we can discuss any questions in the panel. So you should be seeing it in your screens. And the question is, which Lixoft products are you most familiar with, or are you familiar with? And the options are PKanalix, Monolix, and Simulix. So let's give people a couple of seconds to respond before we end the poll. Okay, so most people are familiar with Monolix at about 50%, and the rest is evenly distributed between PKanalix and Simulix. So there's many opportunities coming up to learn more about these products. We have both a page within the Simulations Plus website with previous webinars and announcements of things to come, and there's also a page in the Lixoft website that talks about all the upcoming webinars and training sessions that are going on, and there's plenty of those in the coming few months. And if you're interested in having an analysis done, please contact us at Simulations Plus or Lixoft so that we can help you. I have to say, I love the way that you can look at individual data within the huge plot with all the individuals together, and you can pick out which one is the one that's having a different trend maybe from the rest. And
I agree. This is also my favorite feature in Monolix.
Applying that. Okay, so if there's no questions, I'd suggest we go to then the panel session. Mm-hmm. So for people that have been listening, these sessions for the most part are being recorded and are going to be shared in the future. So you should be able to access them and share them with your colleagues. And for now, let's please go back to the lobby and then log on to the panel session. Thank you very much.
Thank you very much, Amparo.